The gut microbiome is something that researchers are very interested in because of the way that it can impact our health and show markers for disease. There are many reasons why we should be thinking of how to take care of our microbiome, and we will see that there are a lot of ways that a healthy microbiome can influence your health and prevent disease. The absolute definition of what constitutes the microbiome is evolving, but for our purposes, we can think of it as the population of living microorganisms like bacteria, fungi, protozoa, and archaea that exist in various parts of our body, along with their ecological niche or their microenvironment. The major areas where we find these different microbiomes include the nasal passages, oral cavity, the skin, the gastrointestinal tract, and the urogenital tract. Our main focus today will be the gastrointestinal microbiome, which is also sometimes known as gut bacteria. Healthy individuals can house anywhere from 500 to 2,000 species of microorganisms in their gut, with a large majority of those being bacteria, like the ones shown here. The amount of bacteria that someone has is referred to as the richness of their microbiome, and the variety of organisms in their microbiota is referred to as diversity. Diversity of the microbiome is most of what varies from person to person and what we're going to be most focused on. One of the things that draws us to the study of the gut microbiome is its correlation to a bunch of other health markers in different organ systems. The gut bacteria host a bunch of biochemical pathways, which have the very important task of synthesizing vitamins and preparing nutrients for absorption. This has a significant effect on our energy levels and our homeostasis, even in various parts of the body outside the GI tract. Diversity in the microbiome has been linked to many physiological systems because of the various functions that it serves. There are also many correlations of the gut bacteria with disease regarding not only the digestive system, as we would see with IBS and IBD, but almost any other body system that you can think of. It's even been observed that the gut bacteria can sometimes influence mental state and brain function. It's known that the microbiome changes with stress, environment, age, and genetics. But one interesting topic of research is not only identifying the amount and types of bacteria found in the human gut, but also how we can influence the microbiome with lifestyle changes. There are a lot of benefits to overall health that are seen in someone with a healthy gut microbiome, with additional benefits being discovered often. Ways to change or enhance the diversity of the microbiome are being studied, and a large factor of those seems to be diet and exercise. We know about the many benefits of diet and regular exercise, but increasing the diversity of your gut bacteria can be another one of those benefits that come with doing those things. This research paper discusses the differences in microbiota of athletes in reference to other controls that have varying levels of diet and exercise.
We studied a professional rugby team during their time at a preseason training camp to explore the degree to which exercise and diet affects the gut microbial diversity. The rugby team was our experimental group with an average age of 29 years old and a BMI of 29. We included two control groups that were similar to the experimental group in age, but that differed in BMI. BMI stands for body mass index, and it is a weight to height ratio where a value of 25 indicates that the individual is overweight and a value of 30 indicates that the individual is obese. The low BMI control group consisted of 23 individuals whose BMIs were less than or equal to 25, while the high BMI control group consisted of 23 individuals whose BMIs were greater than 28. To confirm that there are differences in the composition and diversity of the microbiota, we knew that the eating habits of these individuals would play a large role, since extremes of exercise are often associated with dietary extremes. The participants were asked to recall their dietary intakes over the previous 4 weeks leading up to the study. Each participant was interviewed by a nutritionist and completed a detailed food frequency questionnaire. This tool was very helpful in allowing us to know which foods had been consumed by which individual and how often they had been consumed. With the help of the nutrition labels printed on the packaging, we were able to successfully record the intake of nutritional supplements by each individual. The manufacturer's weights printed on the packaging allowed us to better quantify this food intake, and we were able to use these findings to make a more accurate calculation of the energy intake per individual. Since the rugby players were involved in frequent, rigorous physical activity, we needed to also assess the physical activity of the members in both control groups. Each participant completed a physical activity questionnaire in which they indicated their levels of physical activities over the past year. This physical activity included activities such as push-ups, running, sit-ups, just physical activity in general. Blood and stool samples were also collected from each participant and they were analyzed. The blood sample was analyzed for protein levels, cytokines, and creatine kinase. And creatine kinase is a common marker of extreme exercise. The stool sample was stored on ice before undergoing PCR amplification. PCR amplification, it allows us to make many copies of the DNA of interest. And the products of this amplification were sequenced, which allowed us to observe the composition and the diversity of the microbiota.
After the different samples were collected, we decided to run some tests. The first test being the Kruskal-Wallis test, which is to determine if an experiment is significant or not. On the x-axis of this graph, we have the different groups that are being measured, which is the athletes, the control BMIs of less than 25, and the control BMIs of greater than 28. And on the y-axis, we have the phylogenetic diversity found within the fecal matter that was provided. The athletes on average had a much higher gut diversity than either of the control groups, with the highest nearing around 200 and the lowest averaging around 50 different species found within the gut. In this graph, we have three different colors that are being shown. The black represents the elite athletes, the red represents the low BMI control group, and the green represents the high BMI control group. In the corners, we have PC1, PC2, and PC3. PC1 stands for principal coordinate 1 and is representing the athletes in this scenario. We can see that all of the black dots, or the majority of them, are down in this right corner with the exception of the outlier. And we can see that the red and green dots are relatively similar across the board. This just shows that the phylogenetic diversity, or the amount of different species found within the gut, was much higher in the principal coordinate 1, which is the elite athletes. And on average, they were about the same throughout the control groups, the different species that were found, but those with a lower BMI had a higher diversity, and those with a lower BMI had a lower diversity. In this graph, on the x-axis, we have the three different groups again being measured, and on the y-axis, we have the creatine kinase levels, which is a marker of extreme exercise. The athletes who exercise a lot more than either of the control groups are more likely to have higher creatine kinase levels, which shows in this graph. And the control BMI of greater than 28, they're not doing much exercise at all, so their creatine kinase levels are a lot smaller. This shows that the athletes had a much higher muscle mass than either of the control groups, which is important in this next slide. In this graph on section A, towards the left, we have again the three different colors, black, green, and red, which are representing the three different groups. Black being the athletes, red being the low BMI, and green being the high BMI control group. Above the x-axis is the majority of the green and red triangles, and below the x-axis is the majority of the black triangles. This is important because this is measuring the different protein that they were consuming and the amounts of protein. The athletes were much more likely to have higher levels of protein intake per day than either of the control groups. And along with this, all three groups had relatively similar types of protein that they were consuming, whether that be white meats or meats in general. But the elite athletes were also consuming a lot of protein supplements, which helped them reach a higher level of protein consumption per day than either of the control groups. These three groups were asked to take a food questionnaire stating which foods they had consumed within the last 4 weeks. So on side B, on the right side of this, we have the different foods that they were partaking and that they were more likely to have. Again, above the x-axis shows the control groups, and below the x-axis shows the elite athletes. It's interesting that the control groups were much more likely to have chocolates and sweets than the elite athletes. They had more snacks versus fruits and vegetables, and they had white bread versus multigrain bread. And then another thing is that they had more alcoholic beverages than the elite athletes, who would much rather preferred sports drinks. This just goes to show that the dietary intake of each of these different groups was different, and that affected their gut diversity that was found. We know that gut diversity is important, and this directly correlates with how much exercise and our dietary consumption. It's important in all ecosystems to have a good gut microdiversity, and exercise and a diversity of dietary needs are helpful in this situation.
There are several mechanisms proposed as to why the microbiota changes in response to exercise, none of which are absolute because research is continually being done. One of these proposals states that exercise changes the gene expression in the lymphatic tissue around the gut, upregulating anti-inflammatory signals and antioxidants while downregulating inflammatory signals. This image just shows the proximity of these gut-associated lymphoid tissues, which is GALT, to the inside of the intestine, which is shown here, to get an idea of how close the inside of the gut and the immune system are, and that they really can influence each other heavily. Another proposed mechanism for why this happens is that exercise affects metabolic flux, which is the rate and direction of different biochemical pathways. Exercises causes skeletal muscle to release different metabolites, and these substances circulating in the blood can interface with the digestive system through immune system pathways, kind of like how we saw in the previous slide. Along with these metabolites, exercise also releases lactate in the blood, where it could alter the gut pH. Although the mechanisms for the effect of exercise on the gut are unclear, there is an obvious connection between the two, which is important to be aware of. Even though this study happened to have focus on elite athletes with intense training and extreme diet, it's important to note that there have been many other studies that suggest that exercise alone in the general population can increase the diversity of the gut microbiota. It was shown, however, that in lean individuals, the microbiota was much more responsive to an exercise regimen than in people who are overweight or obese, which just provides another reason to have a healthy diet and exercise regularly. In our study, we saw that even in the control group with the healthy BMI, the gut diversity was significantly lower than that of our athletes because they were less active. There are many reasons why any given person should include exercise and a healthy diet into their life, but the effect that these can have on the gut microbiome can be extremely impactful to our overall health and longevity, where we would all be able to live longer and healthier lives.